Adverum Biotechnologies is a gene therapy company that's committed to discovering and developing new therapies that will offer life-changing benefits for the patients with limited or burdensome treatment options while living with diseases of the retina, rare diseases, and other ophthalmology diseases as well. And we are gaining momentum in gene therapy. We are doing this through a combination of our technological foundation upon which we have expertise and applying those to opportunities that can help patients. Our foundation as a gene therapy company comes with our vector platform. So again, for gene therapy, most simply, it starts with a therapeutic piece of DNA that's inside of a protein shell called a vector. That vector acts like a Trojan horse to help to carry the DNA into the target cells of choice. The target cells then use their own machinery to read that DNA and to express the protein of interest. So as a gene therapy company, our vector technology platform, we have both naturally occurring serotypes as well as novel variants that maximize and optimize protein expression. Upon this foundation, we have expertise in supply chain, process development, manufacturing, and product quality to ensure safety, purity, and potency of our products. We apply this to a pipeline and opportunities where we can help to meet patients' needs, where there is a clear unmet medical need and validated targets. We have a strong cash position and strategic partnerships with companies like Regeneron and Editas, who recognize that we are uniquely positioned to successfully deliver gene therapy treatments to patients. Here is a snapshot of our lead and partnered programs. Our lead programs have indications for wet macular degeneration, as well as alpha-1 antitrypsin deficiency and hereditary angioedema. For purposes of this presentation, I will focus on our wet macular degeneration program. This audience is well familiar with wet macular degeneration as the leading cause of visual deterioration and legal blindness in individuals over 60 years of age, affecting approximately 1.2 million people in the United States alone. It is the hallmark of wet macular degeneration is choroidal neovascularization and abnormal blood vessels that bleed and break in the macula, impacting vision. VEGF is the main driver of choroidal neovascularization, and regular treatment with anti-VEGF proteins every 4 to 8 weeks can do a good job of controlling that that choroidal neovascularization. However, Medicare data show us that compliance with such a regular treatment regimen is difficult. Further, multiple studies indicate that when there's deviation away from those regular treatment intervals, then that's when there is a reduction in the visual outcomes. So in the real-world setting, there is de facto undertreatment of patients. Gene therapy offers the promise of long-term anti-VEGF expression in the eye after a single administration, thereby addressing the unmet medical need and maximizing patient outcomes. There are a handful or so of gene therapies that have tried to address this need, and the majority of those have been done with a subretinal injection. And that's because of the limitations of the natural variants of vectors, that when you inject them into the intravitreal space, they do not get sufficient protein expression and sufficient retinal cell transduction. That is why we were very encouraged by recent data that we announced having to do with two of our candidates, ADVM-022 and ADVM-032, that were specifically designed to express to express the standard of care anti-VEGF proteins after an intravitreal administration. We studied these two drug candidates, ADVM-022 and ADVM-032, in the industry standard non-human primate laser CNV model with 9 laser spots delivered to each eye, and then those laser spots are graded with fluorescein angiography, with the highest grade, grade 4, representing unequivocal choroidal neovascularization. The number of grade 4 lesions was counted at day 14 and day 28 after after laser, in the eyes of the monkeys who received intravitreal administration of vehicle, standard of care anti-VEGF protein, or R2 candidates. A single intravitreal injection of ADVM022 or ADVM032 demonstrated comparable efficacy to standard of care anti-VEGF proteins in the ability to control choroidal neovascularization. Additionally, pharmacokinetic analysis of these studies showed that we had sustained anti-VEGF protein levels in the vitreous and in the retina for 20 weeks after a single intravitreal injection. So again, the ability to reduce or to control choroidal neovascularization to this effect, and with this sustained anti-VEGF protein levels in the retina and vitreous after a single intravitreal administration of a gene therapy product has not before been demonstrated. Is the direct result of our efforts in vector and capsid optimization, and it differentiates us from the competition who are using subretinal injections that are more complex and more invasive. And it highlights the one-and-done potential of these candidates to minimize the treatment burden of frequent injections and to maximize clinical outcomes. Now, the next steps after such encouraging results are, of course, to move to the clinic. We will be preparing IND, and we'll be initiating pharmacology and toxicology and biodistribution studies shortly. Additionally, we continue to evaluate the durability of the anti-VEGF protein expression beyond 20 weeks. But as I said before, we're a gene therapy company at our core. And so while this is very encouraging data for us, this is only part of the Adverum story. We continue to focus on manufacturing with in-house capabilities that will ensure that we won't be limited in terms of product supply throughout the product development life cycle. We additionally continue to emphasize the discovery of novel vectors and expression cassettes to improve on existing technology and optimize protein expression both systemically and in the eye. We have collaborations that leverage our leadership position in the ophthalmologic vector development. And we have a very strong cash position with existing cash expected to be sufficient to fund lead programs through meaningful human proof of concept data and to continue the momentum as we advance toward the finish line. Thank you.